Hello everyone, my name is Xingfeng Shao from University of Southern California. Today I'm going to talk about our work, comparison between blood-brain barrier permeability to water and gadolinium-based contrast agents in an elderly cohort. And I have nothing to declare. So here's a diagram of the blood-brain barrier. It has multiple layers and starting with a tight junction between the endothelial cells and then parasites and also an outer layer of the astrocyte and feet. So transendothelial permeability is actually very low with a fully functional BBB in healthy brain. And the BBB becomes increasingly permeable with advancing age and also particularly in patients with vascular dementia or cerebral small vessel disease. And DCE MRI with gadolinium-based contrast agents has been widely used to study a broad spectrum of central nervous system disorders associated with BBB disruption. And here, a figure showing actually the water exchange across the BBB is not free diffusion as people used to think. Actually, through this water channel, a protein called aquaporin 4. On the right side of this figure, you can see with AQP4 knockout, the water exchange rate becomes much slower. And recently, study report that the loss of the AQP4 happens with aging, and AQP4 deficiency is associated with reduction in the lymphatic function, and also amyloid deposits has been associated with AQP4 redistribution, leading to a much slower water exchange rate. So the purpose of this study is to directly compare the BBB permeability to contrast agents and also water exchange rate, KW, in a cohort of elderly subjects and risk of cerebral small vessel disease. So we collect our data from the Siemens 3T Prisma scanner, and we use a diffusion prepared arterial spin labeling technique to measure KW and DCE MRI to measure K-trans from the same subjects. For diffusion prepared Picasso sequence, the perfusion signal actually contains two compartments, the capillary and the tissue compartment, and they have a huge difference in diffusion coefficients. So we are able to apply a small diffusion weighting to separate two compartments. And then KW can be quantified using regularized single pass approximation modeling. For DCE MRI, dynamic time series were acquired with contrast agents injection. Then K-trans can be calculated from the signal curve fitting with Patlock modeling, which is designed for low permeability measurements. So here are the imaging parameters for the two sequences. So KW measurements has a relative lower spatial resolution, but cerebral blood flow, arterial transit time, and KW can be quantified simultaneously in 10 minutes. And we use a small p-value of 50 to separate those two compartments. Also, we had test and retest scans about 6 weeks apart to evaluate the reproducibility of the KW measurements. And DCE MR has a much higher implant resolution, but scan time is also relatively longer. So in total, we have 16 subjects, and 50% of those subjects had diabetes or hypertension, but 75% of those subjects has CDR score of 0, which means they're cognitive normal. We also quantify the wet matter hyperintensity lesion using the Fazeca scale, and you can see only 3 subjects has severe wet matter hyperintensity lesions. For data analysis, we first want to evaluate the test-retest reproducibility of the KW measurements using intra-class correlation coefficients. And we study the correlations between the test and retest KW and K-trans using the mixed effects linear regression model in whole brain, gray matter, wet matter, and 6 subcortical regions, including MC perforated territory, caudate, medulla, hippocampus, perihippocampal gyrus, and a combined medial temporal lobe ROI. We also study the KW and K-trans in 3 wet matter hyperintensity related regions, the normal wet matter, the lesion, and its penumbra. So here's the BA plots of test-retest KW measurements in 9 brain areas. And overall, the difference between 2 KW measurements has a very small bias, but the spread of data ranging from about 30 in larger ROIs to almost 50 in smaller ROIs. So the ICC is also pretty good. It's ranging from 0.75 to almost 0.8 in larger brain ROIs, but it's only about 0.4 to 0.6 in smaller brain areas such as hippocampus. So here are the scatter plots of the K-trans versus test-retest KW in 9 brain areas. And the major finding of this study is we found significant correlation of 2 measurements in wet matter, MC perforated territory, and in caudate. And for wet matter, the vasculature supplying the deep wet matter has a very long path, and it's a junction between several vascular territories. So the endothelial cells in wet matter is very likely to be damaged in small vessel disease, which also leading to the wet matter hyperintensity lesions. And for the MCA perforated territory, which is fed by the lenticular striated arteries, which are the end arteries from MCA, with almost no collaterals and also facing a rapid change of the blood pressure. So we found increased tuberosity of those vessels in aged subjects with small vessel disease, which causes increased blood pressure and also alter blood-brain barrier permeability. So we also found significant correlation in caudate, which is consistent with a previous study showing that the BBB dysfunction in caudate is associated with early pathological changes in small vessel disease with minimal AD pathology occurs. So here are the K-trans and KW map from two representative subjects. So on the left, a K-trans map, and on the right is a KW map from test and retest scans. So on the top, this is the subject has a very small white matter hyperintensity lesion, and the second row is data from a normal subject. So first, from the test retest KW measurements, we can see it has a very good agreement between the test retest measurements. But the DCE-MR has a much higher implant spatial resolution. So we did a t-test to see how KW and K-trans changes in 3 different white matter hyperintensity areas. So we found significantly higher K-trans in the lesion penumbra compared to the white matter hyperintensity lesion. So this means K-trans from DCE-MRI could be a useful tool to study the white matter hyperintensity progression. So in summary, DCE-MRI or K-trans measurements is still the most commonly used approach to measure the paracellular leakage of the contrast agents. And DCE-MRI can not only used to detect disrupted BBB, such as in tumors, but can also recently researchers use it to study more subtle BBB breakdown, such as in AD or aging. And overall K-trans in our study, in this study, is very small, but we found highest K-trans in the hippocampus. So this is consistent with the previous study showing the whole brain BBB breakdown starts in the hippocampus. And on the other hand, for the diffusion prepared arterial spin labeling or KW measurements, so there is growing research interest in this. The one reason is the water has a much smaller size compared to gadolinium-based contrast agents, and KW is about 100 per minute in this study, so which is about 5th power of 10 faster than the K-trans. And another reason is the water exchange not only through the passive diffusion, but majority of water go across the BBB through this aquaporin 4, the water channel, which is very sensitive to the lymphatic function. And more specifically, in this study, we found the good reproducibility of the KW measurements, especially in larger ROIs. So we also found significant positive correlations between KW and K-trans, but only in the brain regions that are most susceptible to the vascular impairment caused by a small vessel disease, such as in white matter, MC, perforated territory, and also in caudate. But the underlying mechanism of KW and K-trans are likely to be different, therefore no significant correlations were found in the other brain areas studied. We also found K-trans in the lesion penumbra is significantly higher inside the white matter hyperintensity lesion, so which means the K-trans image have sufficient implant resolution to detect the BBB permeability change in very small ROIs, and can be used to study the disease progression. So I would like to thank my colleagues in USC and also the NIH funding support. Thanks for your attention.